that was I think our last in-class quiz. And then I'm going to give you an out-of-class quiz for the genetics part of the quarter. And we have class on Wednesday, and I'm going to be handing out the review sheet for the final exam. So you'll get your review questions or your essay questions ahead of time. And then we don't obviously have lab this week. So the next time we'll meet back in lab is next Friday for the lab practical.
Will any of the models be in the library at all?
You know, I think I'm going to put a male and a female because I have extra of those. So I'll go put a male and a female model in the library. But I don't have any extra really of the digestive system. Yeah. Okay. So when we're looking at the female's reproductive system, remember that there are two different groups of hormones, and they're released by different structures. So these would be anterior pituitary hormones, the one that is on the top, and the one that is spiking the most would be which hormone? It's actually LH. So this one is LH. And then this one is FSH. Okay. So these have a direct effect on the ovary. And if you look at that spike of LH, you'll notice that corresponds to what happening in the ovary? Ovulation. So it's the spike of LH primarily that causes ovulation to occur. So when we look at the diagram here, you can see that they're showing all of the different oocytes and follicles in the ovary. And so remember that we have at the very bottom here, these would be what are called the primordial follicles. And supposedly we are born with all of those that we are going to mature during our lifetime in our ovary. But during the monthly process, the primordial follicles will then proceed to mature into a primary oocyte. Okay. Now, what distinguishes a primary oocyte from a secondary oocyte? Do you remember? Yes, so the primary oocyte is arrested in prophase 1 of meiosis 1. But if you were just going to look at it in the ovary, what is the difference between the primary oocyte and the secondary oocyte?
The layer around it.
Yes, so the layer. So what happens is that you get a thickening of the layer, the granulosa cells, and then you also start to get a fluid-filled antrum. And so this one would be the secondary oocyte right there. And remember that they said that generally more than one oocyte starts to develop, but only one becomes a mature oocyte, which would be like this one right here in most cases. We're going to talk about twins in a little bit. Okay. So when we look at that one that has that fluid-filled antrum and it has the secondary oocyte in it, that is what is referred to as a vesicular follicle. It's also, when I was learning it, it was called a Graafian follicle, but your book calls it the vesicular. So Graafian is another term for it. Okay. So when the oocyte gets released, notice that it takes some of the cells with it, which are going to form, we're going to talk about the corona radiata in a little bit more detail, but also notice that some of the granulosa cells, or most of the granulosa cells, gets left behind in the ovary. And so these would be granulosa cells right there. And actually also granulosa cells plus that connective tissue layer called the theca colliculi. Theca folliculi? Yeah, folliculi. Okay. Okay. And so what is that yellow structure? It's not yellow on your paper, but what does that yellow structure represent? Yes. So the corpus luteum is right here. So if we look down below at the hormones that are produced by the ovary, so these are the hormones that are produced by the ovary. So you'll notice that there is a surge in estrogen. And that surge in estrogen is due to what? So what is producing that high level of estrogen in the ovary? Does anybody remember? Okay. So that is the mature follicle. So as the follicles mature, like this, they go from primary oocyte to secondary oocyte, and then they become the vesicular follicle. This is the mature follicle. So the mature follicle produces lots of estrogen, right? And notice how that corresponds, that spike in estrogen corresponds with the spike in LH. So when the mature follicle is producing lots, you know, it's ready to be ovulated, and so it actually signals ovulation. So by producing lots of estrogen, so that high level of estrogen causes LH and FSH to surge, right? And that's what leads to ovulation occurring. Okay. So what is producing the progesterone, this high level of progesterone right here? Does anybody know? This is actually the corpus luteum. So these hormones right here, okay, would be produced by the corpus luteum. So the corpus luteum produces estrogen and progesterone. Okay. And then you'll notice all of a sudden it falls off. So this is what happens when the embryo does not implant. And so if you look at that, this diagram right up here, you'll see that the corpus luteum will start to degenerate. So this is the degenerating corpus luteum. So degenerating corpus luteum. Okay. So when the corpus luteum, when the implantation does not occur and there's no HCG or there's not enough HCG, then the corpus luteum degenerates, and then you get a fall or a rapid decrease in estrogen and progesterone levels. Okay. So if we look down here, this is the uterus. Right. So what is causing an increase in the thickness of the endometrium? So this would be the stratum functionalis, and this would be the stratum basalis. So notice that the stratum basalis stays the same. So those are stem cells that are going to give rise to the thickening endometrium. So what causes that endometrium to thicken? Which hormone? Estrogen, right? So notice how progesterone is really low here when this starts to thicken. So this is thickening due to estrogen, right? So estrogen causes the endometrium to thicken. And it does this through mitosis, right? So one of the things about estrogen is it is a hormone that causes cells to divide. And when cells divide, it could potentially lead to a genetic mutation that could cause cancer. And so having excessive levels of estrogen, and also we have estrogen mimicking hormones in, or mimicking chemicals in our environment, like DDT, which is an insecticide, or PCB or dioxin. Those are all industrial synthetic pollutants. They can sometimes mimic estrogen, and so it can actually sometimes mess up reproductive cycles. And it could also be a cause for reproductive cancers in females. So sometimes when there is a risk of reproductive cancers, what they'll do is they'll just take the all the reproductive organs out, right? So they'll give a female a hysterectomy to protect against that, because it could possibly lead to cancer, this continual cell division and cell death. Okay. Okay. So this is a gland. And do you remember what that gland produces? It produces a nutrient for the developing embryo. Glycogen, right? So it produces glycogen. Right? And these are blood vessels. So if the embryo does implant, it will have nutrients, and it will also have a supply of oxygen and nutrients from the maternal circulation. And so those blood vessels are going to play a role when we start talking about pregnancy and the development of the placenta. So you'll notice something that happens here, right, is that if the corpus luteum degenerates and then the progesterone and estrogen levels fall, then we get the sloughing off of the endometrium. So this is what the sloughing off, right, would be due to a decrease in progesterone and estrogen. So most people's female cycles are 28, the average cycle is 28 days in length. And so ovulation occurs 14 days. So if we look at this as being 28 days, right? So this whole thing is 28 days. We would say that this is about the 14th day is when the oocyte will leave the ovary and be ovulated. Are there any questions about that diagram? So remember that it shows the coordination of anterior pituitary hormones and then ovarian hormones, and then what is going on in the ovary versus what is going on in the lining of the uterus. I'm going to start talking about what happens if the embryo does implant or can implant. And so we're going to talk about pregnancy and gestation. And in humans, this is about 280 days, which is about 40 weeks, right? But there's a range. So generally it's between 37 and 42 weeks. Now, gestation is measured actually from the day of your last period, because it's really hard to tell when a female is ovulating, right? So ovulation in humans is hidden. It's really hard to tell. We do have, you can go to the grocery store and buy ovulation tests to tell when you're going to ovulate. But generally, we don't know. Sometimes we feel it. So sometimes when the egg is released from the ovary, you can feel a little pain on one side or the other, on whichever ovary is ovulating. But generally, it's hidden. And so it's measured, pregnancy is measured not from ovulation, but it's measured, or fertilization, but it's measured from the day of the last period. And so what that means is that it's like 2 weeks longer, right? Than because if you look back at that menstrual cycle, right? If we measure when we get our period here, right? And there's 14 days to when we ovulate, then really by the time the egg ovulates, we're actually 2 weeks pregnant already, right? So it's a little bit longer, but it's just the convenient way to measure it, because that's what we can remember. Okay. So when we look at the pregnancy, we can talk about the different stages. And so at 1 week, they call the embryo the conceptus. Okay, so I'm going to write that down. 1 week is the conceptus. Okay. Then between 3 weeks and 8 weeks, so it could just be 3 to 8 weeks, we have the embryo. actually it would be, and then after 9 weeks, I believe it's called 9 weeks. It's called the fetus. Let me make sure that's right. Yeah, so week 9 to birth is called the fetus. So again, fertilization has to take place in the uterine tube or the oviduct. And so it's going to take a little bit of time for implantation to take place. So implantation generally occurs within, let me see if I can find my notes here, 6 to 7 days. So 6 to 7 days, we have implantation. So those 6 and 7 days, that would be that conceptus, which is just really just floating around in the female's reproductive tract prior to being implanted. Okay. So if we look at how we would have to have or coordinate copulation or intercourse in order to get pregnant, we can talk about the viability of the oocyte. So if we look at the oocyte viability, it is viable 12 to 24 hours after ovulation. So what that means is that fertilization has to take place within that early window. So fertilization has to take place within 1 day. Okay. And then if we look at the sperm viability, it is 24 to 28 hours after ejaculation. Okay. So in order for fertilization to take place, sexual intercourse must, or this is sometimes referred to as coitus, so that would be sexual intercourse, must occur no more than 2 days before ovulation or 24 hours after. So as I mentioned before, you can now test for when you are likely to ovulate in order to ensure pregnancy. But there's also, when we talk, we'll talk about birth control, but there's also that idea of natural rhythm birth control, right? Which if you could figure out when you are going to ovulate and you avoid intercourse during that period of time, that would be a way to help to prevent or prevent unwanted pregnancies. Right? Maybe not a very good way to prevent it, but it is one way, possible way to prevent it. Okay. It's not as reliable as some of the other methods. Okay. So let's talk about what has to happen in order for fertilization to take place. So remember that the sperm is ejaculated into the vagina, and then it has to get through the cervix. And so we have what is a cervical mucous membrane, right? So the cervical mucous membrane must thin, right? And it does this because of estrogen. So high levels of estrogen cause that mucus to become thin enough so that the sperm can actually swim through. So this is accomplished during ovulation by high levels of estrogen. Okay, so this allows the sperm to move up through the cervix and into the uterus. Now, it is believed that the sperm actually use chemical sensing. So they actually, sometimes they refer to it as smell, right? So the sperm can actually smell the egg, right? They're not just moving up against gravity. So they actually are able to detect chemicals that are being released by the egg, and they move towards them, right? So the sperm follow chemicals produced by the egg. Okay. So the sperm is able to quote unquote smell the egg. Right? So it smells the egg. Okay. So if we look at the structure of the sperm, it has a head region, and I'm going to draw the nucleus a little bit smaller, right? And then I'm also going to draw a little bag of enzymes right there. And this is the mid piece, and then this is the tail. Okay. So this would be the nucleus. And remember that it is haploid. It is only containing 23 total chromosomes. This mid piece has special mitochondria. So remember that the sperm has to produce energy in order to swim, and it's the seminal vesicle that produces fructose that provides energy. So the mitochondria are kind of taken in the fructose, and they're using oxidative phosphorylation to break it down and produce ATP. Okay, so this is the flagella right here. And this is part of the cytoskeleton. So the flagella moves and allows the sperm to swim. Now, another structure which is really important is this right here. And this is an organelle in the sperm's head, which is called the acrosome. And this contains enzymes. Because if you remember that the egg actually is surrounded by cells, the corona radiata, and it's also surrounded by glycoprotein, the zona pellucida. And so the sperm actually has to digest its way into the egg to get to the egg. Also, on the surface of the head, I'm just gonna put like these little proteins. So I'm gonna put these little proteins here. Okay, so those are membrane proteins. So the first thing that it has to get rid of those membrane proteins. And on the video we watched, we saw that the sperm actually stop when they get to the oviduct, and they actually rub themselves against, right, the lining of the uterine tube, and that strips off those membrane proteins, okay? So that is what is called capacitation. So the sperm rub themselves against the lining of the oviduct. And this lining of the oviduct has cilia on it, and so the cilia are actually moving the egg too, right? And this removes the membrane proteins. And the reason why these proteins are important is that they prevent the enzymes in the acrosome from being released prematurely, right? So this makes it so the acrosome is now ready to release the enzymes. So the acrosome is now, after it strips off those membrane proteins, is now ready to release the enzymes. Okay. So when it releases the enzymes, this is what is called the acrosomal reaction. So this is the release of enzymes. And so hopefully that does not take place until the sperm has actually reached the egg. And this acrosomal reaction helps to break down the barriers. And so it helps the sperm to gain entry into the egg, right? So it breaks down the corona radiata and the zona pellucida. So remember, the corona radiata is composed of cells, the radiating crown around the embryo. And then the zona pellucida is actually glycoprotein. So it's a protein, and that forms the outer kind of membrane. Okay. Now, one of the things that's interesting about this acrosomal reaction is that the first sperm that gets to the egg is not able to fertilize it. So what has to happen is you have to have hundreds of sperm breaking it down originally, and then one sperm can break through. So the first sperm cannot get through the barriers. Okay. So if we look at a diagram that shows this, right? Here's my diagram. It just shows one sperm, but think about how many sperm were in here, right? And so here you can see that they make their way through the corona radiata, which are those cells, and then they get to the zona pellucida, and then they release their enzymes. Now, we also have to have the right species of sperm, right? So if you think about other organisms, like sponges or aquatic organisms, or even fish, if you think about some organisms that release their sperm and their egg to the outside, the sperm has to be able to recognize the right species of egg, right? And this is what keeps the species separated. So if we looked at this, we have to have the sperm has to produce enzymes and then bind to receptors that are on the surface of the cell. So we'll say that the acrosomal process is where the sperm bind to species-specific receptors on the surface of the oocyte. Right. So in the ocean, there's tons of eggs and sperm just floating around, right? And they have to kind of come together, and you have to have the right species. And so we have that too, right? So it's got to be the right sperm. Okay. And so that process is where the sperm breaks through, it would be referred to as fertilization. Okay. So the sperm can fertilize the egg. And so you'll notice here that the head goes into the egg cell, into the cytoplasm, and remember, it swells. We watched that in the video. And then it fuses with the nucleus, right? So we'll just put the nucleus enters the cytoplasm. Of the oocyte, okay? And then remember that the egg is not quite ready to be fertilized. So the egg, or the oocyte, we'll put oocyte must complete meiosis II before the two nuclei can combine. So the entering in of the sperm signals the egg to complete meiosis II, and then the two nuclei combine to form a diploid cell. So this is actually what is shown here. So here's the multiple sperm. Remember, it takes hundreds of sperm to start to break down this barrier before one will come through. So that coming in of the sperm, this is showing the completed, or the meiosis being completed. This is actually also showing how the polar bodies can actually sometimes also go through cell division. So here we have 2 polar bodies, or maybe there's three polar bodies here. And then this is the nucleus of the egg and the nucleus of the sperm, and then they come together to form a single nuclei. Okay. Okay. So let's talk about blocks to polyspermy. So this polyspermy would be that there are too many sperm entering into the egg, right? So we don't want more than 1 sperm. So if more than 1 sperm fertilizes the egg, the resulting embryo is inviable. And it would be inviable because it has too many copies of the chromosomes. Embryo would be inviable. So it'd actually be a polyploid, which means it would be 3N instead of 2N, right? So it would have 3 copies of every chromosome instead of just 2 copies of every chromosome. So it's interesting in plants, polyploids, and a lot of times in plants, they are like polyploid. They have like maybe 3 or 4 different copies of the genes and it doesn't have an effect, but in animals, it's really bad. So the first block to polyspermy is called the fast block. And the fast block has to do with an electrical depolarization. So we get the depolarization of the oocyte, right? So depolarization just means a change in the electrical charge of the membrane. So the electrical charge of the membrane changes. So just like when a neuron depolarizes or a muscle cell depolarizes, we get a depolarization of the oocyte. So that will initially keep any more sperm from entering, but it's not long term. So then we have what is called the slow block. And the slow block has another name, and it is called the cortical reaction. Maybe cortical, sorry, cortical. Is there A there? Let me look at it. I think maybe there is an A. Yes, cortical reaction. Okay. So this is where that depolarization then subsequently causes the release of calcium, stored calcium ions. So that we'll say that there's stored calcium ions in the oocyte. And they're released into the cytoplasm of the oocyte. So they're stored in the oocyte, but they're also released into the cytoplasm from the reticulum, the endoplasmic reticulum, okay? And then this causes cortical granules to be released to the outside of the egg. And this strengthens the zona pellucida. So it makes the zona pellucida stronger. Okay. So we'll say it strengthens zona pellucida. And this forms what is called the fertilization membrane. So once this fertilization membrane is in place, then no other sperm can get through. Okay, so that is the slow block, but that's long acting. So it's kind of interesting. Early embryonic development takes place within this fertilization membrane, and then at a certain point, the embryo breaks free of the fertilization membrane, okay? And then can implant into the lining of the uterus. Okay, so let's look at this diagram right here. Let's see. I have a picture of that. Oh, maybe I have it. It's back this way. Okay. So this actually also shows the acrosomal reaction and the cortical reaction. Okay. So this is the cortical reaction. So notice that right under the layer of the oocyte, under the plasma membrane, we have these cortical granules. So there's sacs of cortical granules. And so when calcium is released, it causes those cortical granules to fuse to the outside of the oocyte membrane, and then they release this. And so this is the cortical granules being released, right? And then you get the fertilization membrane being formed. And let's see if I have another picture of that. No, I don't. Actually, this would be the fertilization membrane. They're just showing it here, I believe. Okay, so let's talk about early embryonic development, and then we're going to watch the rest of that little video that we were watching on last Wednesday. So when we talk about embryonic development, we start out with a fertilized egg, and this is what is called a zygote. And so the zygote is surrounded by the fertilization membrane. So this is my, oops, let me draw this again. Oh, my pen's not working right. Okay, so this is my oocyte, and surrounding it, not my oocyte, but this is my fertilized egg. So surrounding that is the fertilization membrane. So this is the fertilization membrane. And this is my zygote. Okay. So this will undergo cell division, and the cytoplasm is halved equally. So we have equal cytokinesis. So within this fertilization membrane, then we would have two cells. Oh, geez. I don't know what's going on with my pen. Sorry. So we'd have two cells within this early. It's freaking out on me. There we go. Okay. This early division is mitosis, and it is called cleavage. So that is where the cell is cleaved into two, right? And then the next cell division would give you four. So you'd never have an embryo like with 3 or 7, an odd number, right? So the embryo is going to just develop, continue to develop, and the cells are just going to have. And so we're going to get 4 cell stage. Right. Now, once we get up to 16 cells, we have the stage which is referred to as a morula. Might have to quit early. 16 cells. And this is a morula. Okay. Now what is going to happen is that the cells are going to start to migrate to the outside. So we're going to get an embryo that has composed of many cells. And there's going to be cells on the outside, and these have a specific name. So this is what is called the trophoblast. So these outer cells are called the Maybe I'm running out of batteries. blast. Okay. And then inside, we're going to have what is called the inner cell mass. So the inner cell mass is what is going to give rise I should say mass. That is what is going to give rise to the embryo. Okay. And it's at this stage, this is called a blastula or a blastocyst. Okay. And it's at this stage that the embryo leaves, right? It actually leaves the fertilization membrane behind. And it actually has cilia, and it actually can kind of swim around inside the uterus prior to implantation. Okay. So I'm going to show you the video a little bit early since my pen's freaking out. And so I'm going to back up a little bit to the moment of fertilization. So remember again, these would be, oops, move this over there. This is the oocyte, right? This is my membrane that's surrounding it. These are actually the polar bodies. And so here we see that this is the sperm nucleus. And what I'm looking for is that fast block. So let's see if I can back it up a little bit more.
An amazing effort, but without the cooperation of the egg, it probably wouldn't be enough. The support cells that now make up the wall of the egg are changing shape, weakening the wall just enough to draw the sperm in. The sperm keep trying, the explosive energy of their tails driving them along. Most of the remaining sperm die in this last attempt to penetrate the egg. Finally, success.
Okay, so this is the fast block right there.
Sperm has broken.
That's the depolarization.
Instantly, a burst of electrical activity changes the surface of the egg, making it totally impenetrable. 300 million sperm set out. One made it to the finish line. The sperm continues to move for a short time while inside the egg, and then its tail drops off. The sperm's head slowly starts to swell until it is 200 times its original size, preparing to combine its genes with those of the egg. The two large round structures hold the ingredients for a new human being, half the genes from the woman, half from the man. Now, the moment of fertilization. The two pools of genes appear here as the two interlocking circles. They fuse with immense power. United, they dissolve into the egg. The blueprint for a new individual is set. A combination of inherited characteristics unique in all the world has been created. Almost immediately, the egg divides itself into two identical cells. These daughter cells are the first of the billions to come that will culminate in the birth of a single new human being, or maybe more. Mother-to-be Karen Hatchett-Pavlin is an identical twin. Like Karen, her sister Kim was also once a track star. Today, Kim is an investment banker, and Karen is an account executive for Xerox. Like many twins, Karen and Kim are close. Sometimes they believe they can even sense what the other is experiencing.
It just had a very, very strange feeling when she called. I remember Rob was away, and I just knew she was pregnant. I just knew it was it. And it just convinced me to go ahead and take the test and find out. And she was right. We were really excited. Yeah.
So you didn't program them?
She's not supposed to know that. Being twins, we always wondered if Kim was having this baby with my husband, if the baby would look a lot alike, or if they would look like twins, or obviously they would look like brother and sister, because we're genetically the same. So it's an interesting thing. It's an interesting experiment for maybe not us, but somebody else.
Much of who we are as people is determined by our genes. Eye color, hair color, looks, even some personality traits. And all of this is set in the first moments of pregnancy, when maternal and paternal genes fuse together. In the first few days of pregnancy, the egg cells continue to divide as they slowly float down the fallopian tube toward the uterus. Sometimes, about once every 300 pregnancies, this group of identical cells may divide in half. Identical twins like Karen and Kim are the result. Fraternal twins develop when two eggs escape the ovary and are fertilized by separate sperm. By day 4, the egg finally reaches the uterus. Now a startling change takes place. The cells begin to differ from one another. Within the egg, dark-colored blastocyst develops. This ball of cells is preparing to leave the egg behind and move on to the next stage of development.
So that's the fertilization membrane is going to leave.
In this process, never before seen on film, the blastocyst shakes the egg like a baby chick about to hatch. After a moment, it succeeds in freeing itself. Once the healthy blastocyst is separated from the egg wall, it floats freely in the uterus. Close up, the blastocyst shows its makeup. The light brown cells on the lower right will become the placenta. The dark mass on the upper left, the embryo, which will grow in 9 months into a new baby. Day 5, when the blastocyst finds its way to the uterine wall. Its sticky surface helps it adhere and the blastocyst implants itself. Sometimes the process doesn't work just right, and the body is quick to respond. Here, 3 round chromosome sacs lie inside a fertilized egg. This occurs rarely, when 2 separate sperm have both managed entry to the egg. Because of the extra half set of chromosomes, this egg could never develop into a normal human being. The body recognizes the problem, and the egg is rejected. These early miscarriages often occur so quickly and automatically that the woman hardly has a chance to register that she has been pregnant at all. Day 21, 3 weeks after fertilization. Clear as glass, the developing heart begins to beat. Blood cells appear, running backwards and forwards in newly constructed blood vessels, their thin walls providing the developing tissues with oxygen. At 4 weeks, the heart beats on, ever stronger. The beginnings of a head and face have appeared. This is the start of an eye. The liver takes shape to the left of the heart, the spinal column below. The embryo of almost any animal that has a backbone, from a fish to a tiger to a human being, looks just like this at this early stage of development. But now, in the human embryo, Millions of years of evolution are compressed into a few short months. 4 weeks. The embryo is 4 millimeters long. The arms appear as narrow protrusions. This part will grow into a hand. 5 weeks. Primitive fingers have become apparent. The frog-like webbing between the fingers gradually recedes. Week 16. Hands and face are clearly recognizable. The fetus floats in the uterus, surrounded by a protective fluid sac. This flexible coil is the umbilical cord. The umbilical cord belongs to the fetus, but it is connected to the placenta, the vital link between fetus and mother. The placenta, firmly rooted to the uterine wall, is made up of an astonishing network of blood vessels. From the early stages of pregnancy, everything the fetus needs to survive and grow passes from the mother to the fetus through the placenta. In this animated view, the mother's blood, carrying nourishment for the fetus, passes through tiny holes into the placenta's many chambers. What look like trees inside so many boxes are really the branched endings of the fetus's umbilical cord, the villi. The mother's blood will stay separate from the fetus. So how is the fetus nourished? A closer look reveals the secret. The surface of the villi is made of a selectively permeable membrane, a strainer. It allows nutrients from the mother in, but keeps her blood out. The nutrients seep through the membrane into the fetus's blood supply. This system protects the fetus from some harmful substances, but cannot keep alcohol, certain drugs, and some viruses from passing through. This view shows what the process looks like inside the body. Mother's blood shooting into the exchange chamber and bathing the villi, dangling down on either side of the screen. This animation shows the mother's antibodies surging along with the blood into the exchange chamber. For 9 months, the fetus is protected and nourished in this way. And it grows, readying itself for a new life in the world outside the mother. This is an ultrasound picture of the heartbeat just before birth. Now, 18 hours into labor, the baby's condition is carefully monitored, and Karen's endurance is put to the test.
Contraction begins. Right? Here it comes. Deep breath. Breathe out. Speaking. One more, come on. Stay with it. Jump. Stay with it. One more step.
Okay, so I'll continue on with the chapter on pregnancy and development on Wednesday. And I'll give you your review sheet on Wednesday too. And you guys need to take your quiz. Can you come downstairs to my office?
When you're able to resync the master and stuff again,
Yes, I will.
because that's how I get that I try to keep track of it by doing that.
Okay.
Thank you.